Welcome to lesson 7.9. This lesson is about nematode trapping fungi. As you can see in the image on the left, this poor nematode has been captured by one of the nematode trapping fungi. And in this lesson, we'll go into the various mechanisms that nematode trapping fungi engage in order to ensnare their prey, the humble nematode. Okay, so without further ado, let's begin lesson 7.9. So nematode trapping fungi create specific structures or modify their hyphae in order to entrap nematodes. And these come in different shapes and sizes. But basically, they can be divided into two different types. Ones that actually produce adhesive substances to glue the nematodes to themselves. And these include adhesive hyphae, three-dimensional nets and knobs, versus non-adhesive structures. And these include constricting rings. So the mechanism of trapping and killing the nematode are different in these two different forms of traps. Fungi in the family Orbiliaceae produce 5 trap types, and that includes conidial traps. Their asexual spores are also used as traps. And you can see in the image here below that sticky fungal nets are actually produced in this case to trap the nematode, whereas in this image we have a constricting ring trapping and killing the nematode. So now let's look at the various examples of traps produced by fungi in order to consume nematodes. So for example, the humble oyster mushroom produces these structures that look like lollipops called Toxocysts. And these are filled with volatile ketones called 3-octanone. And they are quite fragile. So when a nematode disturbs these toxocysts, the gaseous 3-octanone is released from these structures, and this causes a rapid paralysis of the nematode via an influx of calcium into the nematode's neuromuscular system. So basically the nematode is rapidly immobilized and killed, and then the hyphae of Pleurotus austriatus, which is our oyster mushroom, grows throughout the nematode and consume it. And this is just a mechanism that Pleurotus uses in order to gain extra nutrients from its nitrogen-poor environment. So all the saprotrophic fungi that have developed various mechanisms to consume nematodes do so because there is a lack of specific nutrients, particularly low nitrogen situation in the soil or in the substrate they live in. And so they use nematodes as an extra nutrient source. So now I'm going to show you some footage titled Trapping of Nematodes by Adhesive Branches of Monacrosporium cyanopagum. You can watch the whole video using this link here. And the video was made through a collaboration between the Department of Microbial Ecology at Lund University in Sweden, and there's also a university in Canada that has been involved, and these are just the credits from the video. And so what I have done is explained the footage itself, but I recommend watching the full video with the original voiceover. So Monacrosporium cyanopagum produces adhesive branches of sticky trapping hyphae, these thick, short, stubby hyphae of fungal cells, and they look very different to the usual hyphae. And here we can see just how much they differ from the normal elongated hyphae that they grow out from. These adhesive branches can actually grow into one another, and this is called anastomosis. And they can actually produce these ring-like, lasso-shaped structures as a result of anastomosis. And as the nematodes move past them, they trigger the response of these adhesive hyphae, these sticky branches. So it takes a little while for the branches to be activated. And once attached, it's impossible for the nematode to actually detach itself from those sticky branches. And here we have a Phalangus eveni, our famous fungal feeding nematode, which is being trapped by one of those sticky traps. And you can see that only 2 cells of that branch are actually attached to the nematode. There's also a juvenile that's trying to actually consume the sticky branch, but it cannot penetrate. But at the same time, it can also not be trapped by any of these cells, which is interesting. And you can see here just how much the nematode is pulling on the hyphae. And this time-lapse shows just how strong this adhesion is. And no matter how much the poor creature tries to break free, it is just impossible to do so. And notice the very rapid cytoplasmic activity within the trapping cells here as well, as the fungi work their magic in paralyzing the nematode. And once the nematode is paralyzed, inside the nematode there are digestive hyphae, and the enzymes within the hyphae then work to dissolve the nematode in order to absorb its contents. And then you can see how these new trapping hyphae grow out from the nematode as it's being digested. Once the adhesive hyphae firmly attach, the next step is the formation of an infection bulb inside the nematode. And this is occurring an hour or so after the first contact has been made between the trapping hyphae and the nematode. And here you can see the infection bulb developing as it swells up inside the nematode and then grows into a nutrient-providing hypha that grows throughout the nematode and dissolves the content of the nematode and absorbs the nutrients that result. And here you can see the hypha growing within the nematode. And this is a time lapse over 36 hours showing a complete digestion of the nematode and the proliferation of the hyphae of Monacrosporium. So first the animal was paralyzed, and then the nutritive hyphae are actually taking over the animal, lysing the entire animal, so digesting it. And then the nutrients from the digested nematode are basically transferred or sucked up by the hyphae. As the nematode is disintegrated, all of the contents of the nematode basically is used for the purpose of creating more and more hyphae. And this allows for rapid development of the fungal mycelium. There are some fungal species that actually create constricting rings to trap their nematodes. And as you can see here, the nematode has to go through the loop that the fungi create with their hyphae, and this is the constricting ring. And if the nematode is able to trigger the response within the fungi, they will actually constrict to trap the nematode and kill the nematode, and then once again enter the nematode and consume its contents. So these are micrographs showing the constricting ring pumped up with liquid to trap and kill the nematode. So just to contrast the nets that are formed by some fungi that do not use constricting rings, we have an image here on the left, and basically these are just hyphae that anastomose and grow into one another to create these sticky nets within which the nematode gets trapped, whereas these featured here on the bottom left and in the electron micrograph on the right are actually the 3-celled rings that get triggered and pump up with liquid to capture, constrict, and consume the nematode. And so this is an open ring here on the left, and in this electron micrograph here on the right, we can see that these cells have now swelled up and closed off and constricted the ring. So if a nematode is in there, it would be completely trapped by this mechanism. So a well-studied species that creates constricting rings is Arthrobotrys broccopaga, and the mechanism of trapping is activated when a nematode enters the ring structure, but also heat has been shown to trigger an irreversible ring closure. And this happens via the rapid increase in cell volume. And here we have a ring swelling up, and this is what it looks like when it's fully closed. And this is when the nematode has been trapped. Now we'll see this in action via a video called Trapping of Nematodes by Constricting Rings of Arthrobotrys dactyloides and Dactylaria broccopaga. And here is a link to that video so you can watch the full footage. And these are the credits from the end of the video. And basically, once again, I've just taken some parts of the video and put my voice over it, but I recommend watching the original at this link. So here is a nematode trapped by a constricting ring of one of the species of fungi that creates these rings. So these are the ring-shaped trapping structures of Arthrobotrys dactyloides. And they are sort of intermittent along the hyphae and formed in groups. As to whether a nematode will be trapped by these rings depends on several factors, such as the diameter of the nematode in order for it to trigger the trapping mechanism. But once the fungi detects the nematode and the trap is triggered, the ring will start to tighten around the nematode. This poor nematode has been caught by the neck. So the traps actually begin as a hypha, and then the three terminal cells form a ring. So these are rings of Arthrobotrys dactyloides with a sort of a triangular shape, whereas Dactylaria broccopaga has rings that are larger but also rounder in shape, but the rings always have three cells as part of their structure. Besides being mechanically triggered, there's also heat detection in these rings, which triggers the trapping mechanism. And once triggered, there is a rapid inflation of the three cells, creating a constriction within the ring. And this is slowed down footage by 4 times than the normal speed of the contraption closing. And you can see that the cells are actually separated from one another. Each of the cells has a separate capacity to swell up, as shown here. And this electron micrograph shows that the outer portion of the cells, the outer walls, do not stretch. They remain constant. It is the inner parts of the cells that swell up. So once the nematode stops fighting and starts to die, the penetration peg will develop and the hyphae will start to grow throughout the nematode. And this swelling of the cells creates a seal around the penetration point. So therefore, the contents of the nematode is maintained within the nematode, and this prevents it from leaking out. And this is a fast time lapse of the infection process where the hyphae start to grow within the nematode, and the animal actually dies within about an hour. And the hyphae are basically enzymatically digesting the contents of the nematode and then absorb the nutrients that result. And then within 7 hours, there is enough nutrients in the hyphae and they start to emerge from the nematode. The whole process actually lasts around 2 days between when the nematode is first entrapped to the digestive process and conversion of the nematode's body into hyphal growth or complete digestion of the nematode. So before a nematode is trapped, there is a whole process that goes on that involves, first of all, recognition and then adhesion. So in the species of fungi Arthrobotrys oligospora, there are specific sex pheromones that the hyphae produce, mimicking those of the nematode, as well as chemical food cues are produced to attract the nematode. So there is a lure before this interaction occurs. Then the process of recognition involves fungal lectins, proteins, and nematode sugar molecules on the cuticle. So there's basically a biochemical interaction that needs to occur before adhesion occurs. And upon adhesion, the trap cells make contact with the nematode, as well as further communication occurs between the two parties. So initially, Arthrobotrys oligospora is a saprotrophic fungus. So basically it just feeds on organic matter in the soil. But there are special volatile organic compounds that are released, such as furanone, pyrone, maltol, and nitrate, as well as autophagy, so disintegration of the fungal cells at a time when the traps are forming. And the fungus switches between being a saprotroph to becoming a predator, and this then causes the adhesion to occur. Then a penetration tube is formed, and this results in mechanical pressure upon the nematode cuticle, as well as hydrolytic enzymes are released, such as serine proteases, chitinases, and collagenases, to basically disintegrate the cuticle tissue for the penetration tube to have easier access into the nematode. And then at digestion, the nematode contents is basically turned into lipid droplets. That's a fatty component of the nematode that is then converted into fungal growth and allows the fungus to reproduce. Arthrobotrys oligospora conidia, which are the asexual spores, also form traps. So this is how versatile this species is, and this is the process that it engages upon contact with nematodes. So like all creatures, the nematode trapping fungi have their specific food preferences. So there is a specific predator-prey relationship that is involved. And in this particular study, 12 plant, parasitic, fungivorous, and bacterivorous nematode species were subjected to various species of nematode trapping fungi, which successfully captured most of the plant parasites, as well as nematodes in the family Rhabditidae. Interestingly, they did not prey on Aphelingus aveni, which is a fungal predator. Acroboloides species, which is a bacterial feeding nematode, was also not consumed by the various fungi. So while other nematodes were being consumed, these two species increased in numbers. So as the fungi fed on all these nematodes, Aphelingus aveni was profiting from the fungal biomass that was being developed in this process. So Aphelingus aveni was happy to consume the fungi that benefited from predating on the nematodes. As to why Acroboloides didn't get consumed, that depends obviously on many factors, such as the ability of these fungi to recognize Acroboloides as a potential food source, biochemical signals that are used as recognition and susceptibility cues between the fungi and the nematodes, and obviously Acroboloides was not recognized in this case as a food source for any of the fungi that were involved in the study. So it is interesting in terms of nutrient cycling and acquisition, because the fungi that consume nematodes obviously transfer nutrients into the fungal tissue, but then fungal feeding nematodes end up utilizing those nutrients that originally came from the nematode. So this is just another way that nutrients are cycled within the ecosystem through these nematode and nematode trapping fungi relationships. And in all, that means that basically nutrients are constantly being made available, and there's energy transfer among the different groups of organisms, keeping the nutrients moving and cycling throughout the soil ecosystem. And here we've got a poor trapped nematode and the constricting ring tightened around its body. Another fascinating piece of the puzzle is that bacteria are often associated with nematode-trapping fungi. And in fact, they enlist nematode-trapping fungi to kill nematodes as a way of protecting themselves from being eaten by nematodes. So in this particular study, the researchers have found that 18 genera of cow dung bacteria defend themselves against nematodes using fungi. So the bacteria release urea, which triggers the saprophytic Arthrobotrys oligospora to trap and kill nematodes, leading to complete elimination of nematodes within the vicinity of the bacteria and Arthrobotrys. Bacterial biofilms formed by Stenotrophomonas and Rhizobium, found on hyphae, also have been found to trigger trap formation in fungi. So this is once again another level of interaction that is added to the mix. It isn't just the nematodes and the fungi interacting, but the bacteria get involved for their own purpose. And of course, this always happens in biodiverse ecosystems where there's lots of interactions and lots of different reasons why things happen and lots of mechanisms being involved, both biochemically and physiologically, making relationships between the organisms in the soil extremely complex. And here on the right, we've got a beautiful scanning electron micrograph of an Arthrobotrys oligospora peptide-induced adhesive trap. Okay, so we have come to the end of lesson 7.9, and in our next lesson we will look at the herbivorous nematodes, often also classified as plant parasitic nematodes. So these are nematodes that feed on plants, particularly the roots of plants. In the image on the left, we have the juvenile root knot nematode, Meloidogyne incognita, penetrating a tomato root. So you'll be hearing from me again in lesson 7.10. Bye for now.